Cells are very selective about the stuff they let inside. The plasma membrane is a formidable barrier, and in my last episode I described cell surface receptors, which serve as a way for the cell to choose a desired cargo. I also told you how we engineered vaults to hitch a ride into a cell on one of these receptors. However, once inside the cell, cargo is further restricted, walled off in a membrane-surrounded vesicle. Most stuff that makes its way into these vesicles is treated like food and transferred to digestive organelles called lysosomes. Let's face it, we were interested in delivering drugs to cells, not serving a meal. May I interest you in a delicious vault hors d'oeuvre? They are high in protein and completely fat-free. Hi, I'm Lenny Rome, the Vault Guy. I'm a cell biologist and a nanotechnologist. My passion is a common particle of the human cell called a vault. My daughter-in-law Tara and I are making these lessons to explain what vaults are to non-scientists and teach a little basic science in the process. Our last video described the engineering of vaults to allow them to be taken up by any cell. This was an important advance toward using recombinant vaults as delivery vehicles. Today, I want to cover three things. First, I'm going to describe how some viruses have figured out how to escape the endosome. Second, I'll explain how we measure endosome escape. And third, I'll describe the vaults we made to open the endosome. Stick around. You might be surprised by what happens to a cell when its endosomes are popped. I thought it would surely kill them. We thought about ways to avoid the lysosome, so we studied how certain pathogens, like viruses, had figured this out. A virus wants to deliver its DNA or RNA into the cell's cytoplasm. To do this, many different viruses have figured out how to get to the cytoplasm by hitching a ride into the cell through binding to a cell surface receptor, and then once inside the endosome, they fuse with the endosome membrane to release their DNA or RNA into the cell's cytoplasm. I became aware of a specific viral entry path through my collaborator Phoebe Stewart. Phoebe introduced Val and I to a virologist at the Scripps Research Institute in La Jolla, California, Glenn Nemero. Phoebe and Glenn were collaborating to solve the cryostructure of a virus binding to a cell surface receptor. The virus they were studying, adenovirus, was the same virus I told you about in episode 17 that Steve Dubinet was using to deliver CCL21C DNA into dendritic cells. Adenovirus is about the same size as a vault, and it infects a host cell by hitching a ride on a cell surface receptor called CAR, which leads to its internalization into the endosome, using the process I described in the last episode called receptor-mediated endocytosis. After endosomes form, they can sort stuff coming into the cell. If they contain nutrients, they become more acidic and fuse with lysosomes, where digestive enzymes can act. Adenovirus carries out a clever trick to escape the endosome. As the endosome begins to become more acidic, the virus structure is altered, and a viral protein called protein 6, P6 for short, is released from the virus. Glenn's lab had shown that P6 is a protein with membrane lytic activity, meaning it can punch a hole in the endosome membrane, resulting in movement of the virus into the cell's cytoplasm. Val and I took a trip down to La Jolla to meet Glenn at the Scripps Research Institute. What an amazing place to do research, with a view of the Pacific and the magnificent Torrey Pines golf course. Our visit led to a very productive collaboration where we explored the feasibility of improving vault penetration into target cells by incorporating parts of the P6 protein into the inside of vaults. First, we made a P6int fusion protein and packaged it into vaults. To determine if the fusion protein was active, we carried out a membrane lysis assay. We prepared lipid vesicles called liposomes. Liposomes have a membrane that is similar to the structure of a cell membrane. The liposomes contain a highly fluorescent molecule called a dye or a fluorophore. The brightness of the fluorescence of the dye is quenched, which means lowered, when the dye is packed together in a high concentration. The dye trapped in the liposomes was at a high local concentration and thus quenched. So if we measured the fluorescence, something that's easily done in a common lab instrument called a fluorimeter, the fluorescence was low. However, if a liposome breaks, the dye is released and diluted. The diluted dye now strongly emits fluorescence. More lysis of liposomes gives more fluorescence. A detergent called Triton could be added to lyse all the liposomes to measure the complete release of dye. We used this assay to measure breaking of the liposomes by P6, the gray bar, the P6int fusion protein, the green bar, and the vault packaged with the P6, the orange bar. Interestingly, the packaging of the P6int into a vault did not prevent the lytic activity. These experiments suggested that vaults are dynamic and breathing in solution, thus exposing P6int to the liposomes. We next moved on to measure if the P6int vault could open the endosome membrane. We did this by using a clever assay that was developed in Glenn's lab to show how adenovirus escaped from the endosome. A small circular double-stranded DNA molecule called a plasmid was added to the cells with the adenovirus. This plasmid carried the gene for the green fluorescent protein. Although the plasmid couldn't get into the cell cytoplasm by itself, it was at a high enough concentration that it was able to slip into the cell with the virus, a process that Glenn called the bystander effect. Once the virus opened the endosome membrane, the plasmid got into the cytoplasm and made its way into the cell nucleus, where it could be transcribed into mRNA, exit the nucleus, and be translated into green fluorescent protein and turn the cell green. Using this assay, we could monitor the uptake of vaults into cells and see if our vault engineered to contain P6 could open the endosomal membrane. Without any vaults, less than 2% of the cells turned green, and this number did not increase when free, unpackaged P6int was added. Empty vaults could facilitate the delivery of a small amount of plasmid into the cells. However, the P6int vault allowed over 15% of the cells to be transfected by the plasmid. These findings indicated that vault particles can be modified to open the endosome. To see if delivery could be improved by targeting the P6int vault to cells, we tried the vaults we had engineered to display the growth factor EGF on their surface. These vaults were much better at opening the endosome. Glenn later mapped the P6 domain, and he found that just 20 amino acids harbored the membrane fusion activity. So we decided to fuse these 20 amino acids to the N-terminus of MVP. We called these vaults P6 vaults, and they were even better at opening endosomes than the P6int packaged vaults. To test these P6 vaults with cells, we used anti-EGFR antibodies to target the P6 vaults to cells overexpressing EGFR. These vaults were packaged with a red fluorescent protein called M-cherry. The vaults were taken up and they opened the endosomes and released the red fluorescence into the cytoplasm. Without the P6, the vaults were not released from the endosome. So, what happened to the cells where nearly all of their endosomes were lysed? I thought they would croak, but that didn't happen. At first, the cytoplasm looked like Swiss cheese with lots of holes or spaces where the endosomes were. But after a day or two in the cell incubator, they completely recovered. Cells are very resilient. I'm Lenny Rome, the Vault Guy. Join me in my next episode, where we will be exploring an entirely new direction, the use of vaults for environmental remediation. As always, please like and subscribe, and I always enjoy your comments on my lessons.